All right. Hi, Abigail.
Hi, Clarissa.
Sorry for the tech issues. My stupid computer died at 30%.
I don't think you could have seen that coming, so it's okay.
Yeah. Anyways, I'm super happy to have you here. And thanks for putting in all the time and effort to making a presentation. I'm excited to talk to you and get to learn
I'm happy
more
to be
about
here.
you.
Thank you.
Yeah. So just to start off, for anyone who is watching and doesn't already know what SSI is, SSI stands for the Summer Systematics Institute. It's run by the California Academy of Sciences here in San Francisco, and Abigail and I are 2 of the 9 interns that they have this summer. I am the science communications intern, and the rest of the interns, like Abigail, are actually doing hard science, lab science. And so, yeah, we get to learn about Abigail and your research with corals.
Yeah.
Yeah, if you want, we can start the slideshow and you can get talking a little bit about just introducing yourself. And yeah.
Mm-hmm. Sure, sounds good.
Let's see.
Okay, so this is my About Me slideshow. My name is Abigail Fritz, as Clarissa said. I'm a 3rd-year biology student from the University of Dallas, and my hometown is Austin, Texas. I'm studying biology with a minor in the philosophy of science, and right now at the Academy, I am working with a set of North Pacific octocorals.
Sweet. Alrighty. And yeah, let's just, kind of do a little bit of a timeline of your life. But looking back at kind of your childhood and some of your earliest experiences, what were some of your earliest experiences with science and nature?
Yeah, so I can't remember a time when I wasn't fascinated by nature and the science that explains it and all of that. I think it's a product of exposure mostly. So my parents put a lot of effort into making sure that I was given the opportunity to experience the natural world and just STEM media. So I grew up going to state parks and natural history museums across Texas, just seeing the beauty of these different ecosystems and forms of life in a variety of different settings. And I have a few examples of that here. So the picture on the left is Enchanted Rock State Park. And this is a giant granite dome. I think it's 800 feet in elevation, just rising out of fields, that was revealed over hundreds of millions of years as the softer rock around it eroded away. So it's this really fascinating geological feature that was formed in this really interesting way. While on the right, there is the Waco Mammoth National Monument, which is a museum built over partially excavated mammoth remains from about 70,000 years ago, that were preserved in a mudslide in Waco, Texas. So both of these, I have them here because they were just so intensely cool to me when I was a little kid, 8 or 9, seeing these for the first time. And I obsessively read about them and got super excited about them. And that curiosity and excitement that I felt is something that just has kind of grown to where I am right now.
Yeah, definitely. You don't have to be a little kid to be excited about these things.
Not.
Oh my God. Yeah, you were telling us about the Waco Mammoth National Monument a few weeks ago.
Oh yeah, I was traumatized by it. They have a video there showing an animation of these mammoths dying as they were overtaken by mud. It's very scary for a 5-year-old.
Yeah.
It had a big emotional impact.
Yeah. But at the same time, it's just incredible to see I mean, I've obviously never seen it in person. I've just seen these photos, but yeah, I would really love to go. It seems really incredible.
It's a lot of fun. It is, definitely.
Yeah. Okay. So we have a bit of, geology, some natural history in there, but how did you get to corals, or what are you researching this summer?
I'm going to talk about what I'm researching now first, and then I'll get into how I got here. So first off, I need to go over some coral basics because I don't think a lot of people know much about them. Corals are marine animals that are made up of these little polyps. So you can see on the screen each of those little circles with the tentacles around them. That's a polyp. That is an individual coral creature that consists of this cylindrical tube of flesh with a stomach in the middle, a mouth/anus at the top, and some tentacles that pull food in. And what you typically think of as a coral is made up of thousands of these individuals, and they all are linked together by these lines of tissue, so you can exchange chemical messengers and food and function as a single organism. What I am studying are octocorals, which are corals that have just sets of 8 tentacles around their mouths. And the title of my project. Oh, the title of my project is okay. It got deleted, but studying the morphological diversity and molecular phylogenetics of North Pacific octocorals. Basically, I'm asking how the octocorals I'm studying are related and trying to answer that question by comparing skeletal structure and DNA. The basic idea is the more similar two animals are, the more closely related they are. So you look at the DNA, the fewer differences, the more closely related, and you can construct this big tree of the relationships between every single one of those animals in the set. So it's like 23andMe, but for corals.
That's so fun.
It is. It's really cool.
Yeah.
And it's also interesting seeing how all of these alternative technologies like DNA fingerprinting for forensics purposes and ancestry DNA and medical research with genetics, it's all using the same basic technology that we're using in the lab here at Cal Academy to study just corals, which is really cool.
Yeah, and I mean, besides just the genetic stuff, we did our whole day in the life with you on Instagram already.
Yeah.
And so anyone who is watching this interview from Instagram will already hopefully be a little familiar with you and some of your work. But you also just get to look at these really amazing like coral skeletons under different types of microscopes.
I do. And I had a picture originally of me using the scanning electron microscope. I deleted it somehow. But I am to look at skeletal characteristics. The pieces that I'm looking at are 1/10 of a millimeter long, around that range. So they're not something you can see with the naked eye. You need a scanning electron microscope to bombard this tiny little object with electrons and create like a topographical map of it that can be imaged on the computer, and you can actually see the characteristics. So I'm getting to, and that's one reason I chose this project, was the opportunity to do this genetics research, to use a scanning electron microscope, and to gain these technical skills, while also working with an organism that I'm just fascinated in already, and I've already worked with in the past, which is corals, because they're super cool.
Yeah, and that leads us into our next question. Yeah. But what got you into corals? You grew up in Waco and Austin. They're not,
I did. I grew up in Central Texas. I saw the ocean as like a little strip in the distance when I was like driving to visit my grandparents, and that's about it. Then in my freshman year of college, I basically emailed every single professor in my biology department asking if they had a research position open, because I was just kind of desperate. And the only person who did was a marine biology specialist. So the project I started working on was processing deep sea ROV footage, remotely operated vehicle footage, taken by NOAA in the Pacific Ocean. And what you can see here in these pictures are stills taken from that video. So these were taken from the musician seamounts at about, I think, 3,000 meters under the surface of the ocean. So it's 12,000 feet. There is no light at this level. So the light that's being shown on these in this moment is the first time they've ever seen it, ever. And the first time it's been shown on their lineage in thousands of years. And I was looking at these just beautiful worlds I had never even thought about. Like, I'd never even considered marine biology before. And there's just this complexity and this ethereal beauty to them. And all of a sudden I was looking at them and I grew to know every single coral by name. And I became familiar with them. And it felt like looking at that footage, like I was returning to old friends every single time. And just the complexity of them and of their life form with that colonial growth, it's just really fascinating to me, and I love it.
That's amazing, Abigail. That's so beautiful.
I love it so much.
I know. Honestly, I think the most like peaceful day I've had at the academy so far, just, I'm usually running around doing different things, but just that one day I spent with you just looking at coral skeletons and like IDing them was so lovely. It's
a very relaxing thing. Yeah. There's a lot of peace in it.
Yeah, it's very nice. But yeah, so, from all of this, you've done plenty of research so far. I mean, you're going into your junior year of college.
Yeah, my junior year.
Yeah, you are a well-seasoned veteran of science so far. But what are your plans for the future, after the summer, after this program ends?
So my plans are pretty basic right now, is just finish my degree and then do research. I don't know what exactly that research will look like. I know that I love what I'm doing here. Like you said, it's being in this space surrounded by these specimens that were pulled up from across the world, from across the world's oceans, from every single depth range you can imagine, every single type of habitat. It's such a wealth of information opportunities for research. So I would love this sort of job. I would love to do field work, maybe doing like population analysis. So seeing why are different types of corals distributed in different areas, or how are corals reproducing, or what controls their genetic, like the pattern of asexual versus sexual reproduction, the timing of those. There's a lot of questions I have. I know that I'll be working with corals, and I know that I will probably be using the same skills I've gained here in that research. And also, I think the biggest takeaway I've had from this program is just having had the opportunity to get into the lab, to work with my mentor, to shape my project into something that is interesting to me personally, and to work with so many people who love their work and are doing super cool things with it, including a lot of activism and working with reducing education inequality and performing conservation work. It's increased my confidence in my abilities as a researcher and broadened my scope of what I could do with the research.
Yeah, definitely. I think the other unique thing about this program is just they're funding a lot of students who are coming from some sort of marginalized background or hold some sort of like, diverse identity. And so it's been really reassuring to have so many higher ups in this program who are running this program, just like reinforcing every single day that we are supposed to be here.
It is, yeah. It helps with the imposter syndrome a lot if it's done good work there.
Yeah. Well, thank you so much, Abigail. Is there anything else you'd like to share with anyone watching?
I think that's it for me. Thank you.
Yeah, it's been so lovely talking to you. I love learning about corals now. I'll never see them the same.
Thank you. All right. Have a good day.
All right. Yeah, you too. Bye-bye.